Morning, everyone. Very warm welcome to the Analytical Techniques in Biotechnology session for today. I think very few students have joined us today. Please confirm if my voice is okay and you can see the screen very well. Only then we can start our session of today. Okay, good. So let's start with a little bit of introduction here. So who am I? I'm Dr. Simranjit Singh. I had my BTech in Biotechnology in 2009 at Shahid Singh College of Engineering and Technology in Punjab, and then I went for my masters at University of Gießen, Eustace Liebig University, where I had my masters in agrobiotechnology. But I switched for my master thesis. It's in Germany, identification of novel endothelial specific molecules involved in the regulations of tumor cells. Then went for my PhD in 2013 at the University of Göttingen at the major hospital of the Göttingen, that is University Medical Center, and I got my PhD topic was under the molecular medicine, redox regulations of protein phosphatases, and ER stress regulations of CTGF in cardiomyocytes. Then, after successfully finishing my PhD from Germany, I came back to India and I joined as a AP at Mata Gujari College in 2018 till 19, where I was teaching various subjects of biotechnology, creating syllabus, coursework, many things. Then I joined last year in July 2020 during the lockdown period time with Edufabrica, and we designed many courses like this. And now I'm having these small courses that we can see here. And also I'm a AP at the moment, Rama University in Kanpur since last year November, and where we are teaching many things related to. So I was teaching analytical techniques there also. It's quite similar to your MBBT techniques, but it's quite different from a little bit. So we will be dealing with that part today. So that's something about me. Who am I? What I'm doing? What is my educational background? What is my experience is all about. So I will put the ball over to you. Just tell me where are you from. Give me some introduction. What are you studying? What are your hobbies? And why did you choose biotechnology or biological life sciences? That what you're studying? Just give me your brief uh, comments on the chat box. Please, students, give your comments. Give us your introduction in the chat box so that we can all get to know about each other. Okay. From where are you doing it, and what are your hobbies, Shrishti Patel, and why did you choose microbiology? Good to hear. Thank you, Angelina Swatsian, Mulya Rao. Thank you very much. Who else have joined? Samiksha, what you want to say? Where are you from? What are you studying? So good, great. Thank you everyone uh, for your comments, for your introduction. Nice introduction. Um, so yeah, these live sessions will be for 2 hours. Due to my time constraint, I try to pack this workshop in a single day. So we will have our class from 7 to 9. Then we will have 1 hour break, and then we will go from 11 to 1. Then I think you have a classes of vaccine, some of you students, vaccine technology. Then we will CS from 3 to 5. So this will be a bit intense, but it will be also interesting to do so. So keep your food, everything around you, and get your notes, your pen paper with you always, and we will go through in details with a lot of virtual workshop also, a lot of learning also in it. So if we see the workshop for these 2 days, this three, usually we make it to 3 hours of two workshops, 2 day workshop, but um, yeah, at the moment I'm currently running a molecular biology workshop, enzyme engineering workshop, analytical technique workshop, and medicinal biochemistry workshop. These are all for the 2 days. So far we are done with the enzyme engineering workshop, and the molecular biology and medicinal is going in the evening from 5 to 7 and then 8 to 10. So that's why, so it's starting from the last Friday, this Friday, and yeah, so I need to manage in 3 days. I was asked, so in that way, so this was the only possible reason. So in this, we will be dealing basically on today topic on electrophoresis. So, and also the basic with the Western blotting, isoelectric focusing, 2D gel page, SDS page. So we'll be dealing with this part, and beyond that, we can also go for chromatography, various kinds of chromatography, ion exchange, HPLC, gel filtration chromatography, affinity chromatography. So we can actually manage, not, we will not constrain ourselves only to these techniques. We can go further beyond these techniques also. So be ready with this. So basically, if we're gonna start with techniques today, so it's pure technique subject, so there is nothing I have to teach you any basics about biochemistry or molecular biology. It's a, because if you are having these courses, analytical techniques in your BTech, you might see that you have to go through wide, like in 5 modules, you might be having 5 units, and you have to go in details with chromatography in 1 unit, spectrometry in 1 unit, centrifugation in 1 unit, microscopy in 1 unit. Yeah, so these are all being divided. But today I will try to teach you all these techniques with proper background behind them and proper learning. So with virtual videos and yeah, practical experience also. So it's 7:30 now, so we still have 1.5 hours. So we slowly starts, and gradually we make to the end. So I would like to start with you all students with the DNA isolation first of all, how we do the DNA isolation. And after the DNA isolation, I will take you to the PCR technique, yeah, and then we will come to the electrophoresis. So step by step, we will go, not just straightforwardly. So for DNA isolation, So we will discuss a little bit about how we isolate and purify our DNA from the nucleated cells. So what are the various sources that we can use for the DNA isolation? That is your blood, buccal cells, cultured cells, bacteria, biopsies, forensic samples, all are possible. And then basically your DNA isolation is divided into three different parts. First, you disrupt your cells, remove the cell membrane by doing sonications or by adding detergent. Then you isolate the DNA by removing the proteins by the addition of a protease into that. Then you precipitate the DNA using ice cold ethanol or isopropanol because alcohol is not soluble in the alcohol, so they will come together, and we found a pellet after the centrifugation. So this will remove the alcohol from the soluble salt. So here we can see we do the cell lysis first, protein degradation in the presence of FABG and proteinase K, and then the binding is done, then centrifuge is done, then the washing is done with the wash over 1 and 2, and then after washing elution is done. Then after the elution, pure genomic DNA is that what we got. So we will learn these techniques in the virtual way soon. So the basic rules that you have to maintain while doing so, that you have to lyse your red blood cells with gentle detergents such as Triton X100, wash cells hemoglobin, which inhibits restriction enzymes and Taq polymerase, and which works on ice to slow down enzymatic process, and wear gloves to protect your samples from you, and autoclave all solutions and store in the fridge except SDS and organic solvents. When you keep all pellets and supernatant until you have the DNA that you are looking for. Then getting to the DNA, how do you get DNA? You first lyse the cells, then you add NaCl so that DNA is stabilized and remains as a double helix too. And we add EDTA, which actually chelates magnesium ions and also is a cofactor of DNAs, which chew up the DNA rapidly. And we also add anionic detergents, the SDS, which will disrupt the lipid layers and helps to dissolve membrane and binds positive charges of chromosomal protons to release the DNA to the solution, so that our DNA could be removed out of your histones proteins. Then we add protease, that is your proteinase K, which digests the proteins, incubate the solutions at the elevated temperature. And we also do the by doing so 56 degrees Celsius to inhibit the degradation by DNases for 24 to 24 hours. Then how we can get rid of the extra protein? You can add organic solvent extractions using equal volume phenol and chloroform in the ratio of 24:1, and then protein at the interface after the centrifugation for 10,000 rpm at 10 degrees Celsius for 10 minutes. Then you precipitate the DNA by adding 2.5 to 3 volume of your ice cold 95% ethanol now, and then leave at minus 20 degrees Celsius overnight. Then you centrifuge at 10,000 rpm for 10 minutes, 4 degrees Celsius. Then you wash this DNA pellet to remove the excess salts. That is your 70% ethanol, and air dry, and then you resuspend into the sterile distilled water, pH with 7.4, and then store at the 4 degrees Celsius at minus 20 degrees Celsius long term. Then quantify the DNA. To quantify the DNA, mainly we use the OD of 260 nanometer, where we get the concentration in microgram per ml with this formula. And if it is at 260 OD, you're getting 50 microgram per ml. Then it's a double stranded. If it's single stranded, then 33. If it is RNA, then it's 40 gram per ml. Then how to find the DNA purity? So mainly the optical density, we take a ratio from the 260 to 280. If the ratio coming out to be 1 points less than 1.6, then your protein is contaminated. If it is more than 2.0, then the chloroform of phenols are contaminated. Then you repurify the sample. Then the summary: sample for DNA extractions. lysis of cells at elevated temperature plus detergent, enzyme and salt buffer, and then removal of cellular proteins, precipitations of nucleic acids with ethanol, and quantitations and purity measurements of DNA. So some would be used for agriculture development, environmental protection, and ensure food security for overgrowing human population. So that's it from this part. Now I will take you to the PCR part. So what is PCR? It's basically once you done have you isolated your DNA and you want particular template to be grown properly. So for in that way we do the PCR. That is your polymerase chain reaction, which could be used for further testing. So the short history of PCR. So in 1983, Carey Mullis has developed the PCR, and then in 85, first publications of PCR by Cetus Corporations appears in the Science, and then in 86, purified Taq polymerase was discovered in PCR. Then 1988, Perkin-Elmer introduces the automated thermal cycler. Then in 89, the big thing came out as a Taq polymerase, the molecule of the year. Taq polymerase, yeah. So we have discussed that we use to amplify a lot of dsDNA molecules with the same size and sequence by enzymatic method and cycling conditions. So the first step that we do in PCR is your denaturation with the temperature 92 to 94 degrees Celsius. You have a double stranded DNA which melts and gives single stranded DNA. Then at 92 degrees Celsius, they become a single stranded DNA. Then we decrease the temperature to 50 to 70 degrees Celsius for annealing. That is, your primer and forward primers could attach to the specific your sequence, and then afterwards we again increase the temperature to 72 degrees Celsius so that Taq polymerase could be attached to the annealed primers, and they start to produce your corresponding sequence with the various help of various D nucleotides, DNTPs. Then these are the products of extension. In the first cycle you will have the same sequence that you're looking for of the same template, and if you keep this repeating this cycle for 35 cycles, then you will have multiple millions of copies of the same sample. So the chemical components majorly that you require is magnesium chloride, buffer, DNTPs, primers, DNA polymerase, and target DNA. So basic requirements for PCR reaction, that is you need DNA sequence of target region must be known. Primers are typically 20 to 30 bases in size. This can be readily produced by commercial companies, can also be prepared using DNA synthesizers. And basic requirement for PCR reaction is thermostable DNA polymerase, example Taq polymerase, which is not inactivated by heating to 95 degrees Celsius. And we use thermal cycler, which is a machine program to carry out the heating and cooling. These are the various applications from sequencing, genetic engineering, molecular identifications, varied applications that we can use for it. This is the instrumentations that we used. Your thermal cycler. This is 4-in-1. This is only single one, and the bottom is the centrifuge actually, light cycler. So that's it. So we have discussed now about your DNA isolation and PCR. So whatever the things that I will be discussing in the class, I will be keep sharing them with you. PCR was it? The end part of this slide has my, yeah. So now I will take you to the virtual level of DNA isolation, how we do the DNA isolation, and
any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotech collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotech's proprietary purifying solution, Prep It, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case, 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case, 14,500 RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples. Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample and mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, Place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution, as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Cap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples.
So that was about DNA. Now we go for the PCR.
Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. "12.5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6. Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F, and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water, a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration, a reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. Press and hold the short button to do a brief spin.
That's the last part.
The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. Cap the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin 3 locus, and hit Proceed. The heated lid should be enabled by hitting Proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
I will stop here these videos now.
In this example, DNA samples.
Yeah, I can share these videos, sure. Now we switch to electrophoresis. So electrophoresis techniques. So we are done with the DNA isolation PCR. Now comes the next technique, that is electrophoresis. So this will be basically, we will mainly go theoretical part in this moment, PPTs, but later on I will switch to some very nice virtual workshops also, so that we can understand them from a practical perspective, from the virtual perspective. So electrophoresis, basically it's a technique in which we are capable of getting differentiate between DNA, RNA or protein based on the electric charge by conducting positive and negative electrodes. So we can define it as it's a migration of charged particles to solutions under the influence of external electric field. So ions that are suspended between two electrodes tend to travel towards electrodes that bears opposite charges. And types of electrophoresis. So these are zone electrophoresis, moving boundary electrophoresis. So zone electrophoresis includes your paper electrophoresis, gel electrophoresis, thin layer electrophoresis, cellulose acetate electrophoresis. As the moving boundary electrophoresis includes capillary electrophoresis, isotachphoresis, isoelectric focusing, and immunoelectrophoresis. So we will discuss about these two different articles here: zone electrophoresis and moving boundary electrophoresis, under which we are having four different categories. So let's start with the zone electrophoresis. So basically in zone electrophoresis, we involves the migrations of the charged particles on the supporting media. So like paper, cellulose acetate membrane, starch gel, polyacrylate are the one that we use. And we apply a potential difference at the end of the strips components in order to migration of our samples And the sporting media is saturated with buffer solutions. Small volumes of samples are applied as a narrow band. So some advantages are that there are biochemical investigations. Small quantity of samples can be analyzed and cost is low and easy to maintain. The general methods of operations. So saturations of media with the buffer, sample applications, electrophoretic separations, and the removal of supporting media. So these are the general operation that we do under zone electroforces, and the instrumentation that we use in most of the cases is electrophoretic chamber is required, electrodes, diffusion barriers, and supporting stabilizing media. So first thing first, like paper electrophoresis, which is quite common. But usually we don't use in the lab. Mostly in the lab we are going for SDS page, your western blotting, southern blotting or northern blotting if you're interested in RNA. So these are the basic techniques that are highly used in the lab. But these are also sometimes being used like capillary electrophoresis, isotech but usually they are not highly demanded in the field of molecular biology or analytical techniques in biotechnology. So, but still, they are quite important from the subjective point of view, from your um, examination point of view, because many questions are being asked from these perspective also. So, in that sense, what we can see in this diagram, we are having a 45-volt DC battery and the electrolytes on the left and right with the positive and negative electrodes, and we have these two glass plates in between. We are having this paper. Um, for the paper electrophoresis, and then this could be of two type, horizontal and vertical. So in the horizontal we are having filter paper strip, and in the vertical we are having this diagonal, this triangle uh, thingy. So in this, what we do, we have a positive electrode one end, negative electrode another end. We have a airtight housing, and we have filter paper strips, buffer solution, like this. Where in the vertical paper electrophoresis, we're having these electrodes being attached. Sample application point is there. Then there's airtight housing in between, and then yeah, there's a buffer solution. And you things, we have a paper here, and on this side our paper is here, filter paper strips. So things start to transfer from yeah, negative to positive like this. So we use a filter Wattman paper, such as Wattman number 1, number 3. There's a size, various sizes of them, which has a good effect. And the separations in them, it's take a lot of time. That's a quite disadvantage. 12 to 14 hours. It's quite easy. Doesn't take so much time, or sorry, money. Cost is not so much, but yeah, it's easy to use. But sometimes there's some disadvantages. Electroosmosis could happen there. This is happening in most of the cases. Then there is a second one after the paper electrophoresis, that is your continuous electrophoresis. In the continuous electrophoresis, what we can see, we are having this sample reservoir. Um, so we put a buffer solution on the top and our sample components at the bottom. So with the help of this continuous electrophoresis, our paper, our things could be separated into different compartments with the help of filter paper. And this is the positive electrode and this is the negative electrode. So this is used for preparative scale purpose. Predetermined sample volume of 0.2 ml per minute, and silica gel, powered glass, sand, agar gel can be used, and potential difference of 500 volt is applied, and pure compounds are collected in separate containers. And after complete electrophoresis, the filter is removed from apparatus and stained to locate the separate components. So we are done with the continuous electrophoresis, paper electrophoresis. Now third comes your under the zonal electrophoresis, your gel electrophoresis. So gel electrophoresis mainly brought into the main principle is behind is molecular sieving technique. So in the gel we are having these molecular sieves, small holes through which our smaller particles, bigger or middle size of a protein size or DNA or RNA would be differentiated, would be separated from each other. Gel is quite colloidal in nature in solid form, 99% is water in them. And it is important that supporting media is electrically neutral also. And for the gel, we can use agar gel or Agarose, starch, Cefadex, polyacrylate gel. So these are various gels are also possible. And the porous gel that act as a sieve by retarding or in some cases completely obstructing the movement of macromolecules while allowing the smaller molecules to migrate freely. So A is your casting tray for making the gel. Yeah, in the B we have Agarose solutions poured into the casting tray, and C is the comb that forms wells for the samples, and D is the wells that can be loaded with the samples. So like this, and then we put them our sample in them, and then we put the power supply, and on the based on that our molecules, our DNA or RNA proteins could be separated from each other. Then in the gel electrophoresis, it's carried out in two methods. It could be also be vertical and horizontal. Um, so beyond that, what should we use, agar or Agarose? If you're using agar, it's actually a mixture of polysaccharides extracted from the seaweeds, and it's a quite highly purified uncharged polysaccharides from agar. And whereas the Agarose is a chemically basic disaccharide secreting units of 3,6 anhydro L-galactose. And Agarose dissolves when added to boiling liquid, and it remains liquid until the temperature is lower to 40 degrees Celsius, which points it gels. So Agarose gels are quite like could be broken very easily, as they are hydrocolloids and they held together by formation of weak hydrogen bonds and hydrophobic bonds. So in a sense, when you're using for your gel, better is to use agar gel than the Agarose gel. So some disadvantage, as I said, electroosmosis resolution is quite less as compared to polyacrylate gel in the grass, and the, the advantages are that resolution is superior to the filter paper than the previous things that we are seeing. Sharp zones are obtained. Recovery of protein is good. Easy to prepare. Small concentration of agar is required, so not so expensive. And beyond that, it's used in also in immunoelectrophoresis. So this is what happening to the Agarose. We're having this solid state gel here, and then you make it to around 45 degrees Celsius, and then initial gel looks like this, and then after some times final gel structure would be more complex and make a proper shape. But if you make a temperature of 100 degrees Celsius, it could go reverse for the initial gel and in the solid state afterwards again at 100 degrees Celsius. So let's see how the Agarose gel electrophoresis is done in a
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
I hope this Agarose Electrophoresis is clear. Yeah. So we continue with another, the same thing. Now, further within the same of zone electrophoresis that we are discussing since morning, um, we have discussed paper electrophoresis, gel electrophoresis. So within the gel electrophoresis, we are discussing various types, discussing its importance. Now it comes the next part, that is your polyacrylate gel electrophoresis, that is your page. So it is prepared actually by polymerizing your acrylamide, that is your CH2, CH, NH2 monomers in the presence of methylene bisacrylamide to cross-link the monomers. The structure that we get held together by covalent cross-links, and polyacrylamide gels are tougher than the Agarose gels. So it is a thermostable, transparent, strong, and relatively chemical inert. And gels are uncharged and are prepared in variety of pore sizes. And proteins are separated on the basis of charges to mass ratio and molecular size, and phenomena is called molecular sieving. And the gels are quite stable over a wide range of pH and temperature, and gels of different pore size can be formed. And sample and separation speed is quite good comparatively. There are types of pages. So page could be classified according to the separations conditions into native page, denatured page, or STS page. So in the native page, basically we run in the non-denaturing conditions. We don't have any agents which will denature our protein. So the analytes nature substance is maintained, and separation will be based upon the charge, shape, and size. And this was the first earlier original mode of electrophoresis. Then came the later part, your denatured page electrophoresis, that is a SDS page, so which is a separation based upon the molecular weight of a proteins. And in this we can determine the molecular weight of a protein, and very useful checking the purity of your samples, how much pure is your samples, whether you get a dark band or light band. So based on that we can tell about the purity of your samples also. So in the right hand side we can see we have a positive and negative electrode of the polyacrylate gel, and we are having a large high positive charge, large low positive charge, and the small high positive charge and small low positive charge. Yeah, so they all get separated. So the one with the large positive charge will be here, and the small one with the high positive charge will be here, and in between uh, yours your large high positive and a small low positive will be here somewhere. In page, what we do is basically we will discuss about this in later in the Western blotting. Is actually the part of other Western blotting. It's also called as a page. So we will discuss this in details in one of the later lectures. But in nutshell, I can tell. So you should put your gel with a negative and positive buffer with a high voltage power supply. You have a slab of plates. In between, you put your gel. It's a vertical way, and you put your samples in the sample wells, and then you have proteins could be visible after staining them, and you can visualize with the primary and secondary antibody that whether your protein is present or not, what are the various readouts you want to see out later. So you can visualize these SDS page with the help of ethidium bromide, silver, composite blue dye, and if the analyte molecules fluorescence under ultraviolet light, an autoradiogram can be recorded of the gel also. Then SDS Polyacrylate Gel Electrophoresis, sodium dodecyl sulfate polyacrylate gel electrophoresis. It's a technique widely used in biochemistry, forensic genetics, and molecular biology to separate protein according to the electrophoretic mobility. When a detergent SDS added to SDS page, its combined procedure is termed as SDS page. An SDS code proteins molecules giving all proteins a constant charge mass ratio, and due to masking of charges of proteins by a large negative charge on the SDS binding with them, and the protein migrate along the gels in order of increasing sizes or molecular weights. So what does this SDS do? In the page we just add SDS. So what happened? In the native protein is the unfolded states, yeah, but by heating in the presence of DDT and beta mercaptanol, they get unfolded, and this SDS with after heating and SDS and reductants, your native protein will be single like linear, and they will SDS will give a negative charge towards it. So it will be very easy for us to differentiate between your proteins, and they migrate very easily from your negative electrode to positive electrode. So as we can see, your SDS denature sample with sodium dodecyl sulfate, their SDS are coded. So SDS is basically the ionic detergent that binds to stoichiometric to number of peptide bonds that separates on the basis of a molecular weight, and then place mixture of proteins on gel, apply electric field onto that, then you stain to visualize separated bands later on. So like 1, 2, 3. Then you can also use in the instead of Agarose Gel page, SDS page that we have discussed in electrophoresis, we can also use starch, which could be boiled in buffer to give a colloidal suspension. Suspension on cooling sets as a semi-solid gel due to the intervening of branch chains of myelopectin. In order to avoid swelling and shrinking, petroleum gels are used. Advantages are that they are quite high resolving power, sharp zones are obtained, and components resolves can be recovered in reasonable yields, especially proteins. And they could be used for analytical as well as preparative electrophoresis. Disadvantages are their electroosmotic effects, variation in pore size from batch to batch. And within the electrophoresis, we have discussed that there are two types, vertical and horizontal. So in the top, we are seeing the vertical, and the bottom is the horizontal one. So the same way, like we discussed the paper electrophoresis, in that we have paper, but in this case, we are having a bit different compartments, but we are using gel in them also. So we are using starch gel in this vertical case, and in this case, we are using for petroleum jelly also, gel also, trough is there, and that's how we are running our things. So sample is mixed with the gel, and we soak this bit of filter paper in the sample, pressing into the gel, and we detect the staining by UV absorption later on. So that was all about your gel electrophoresis, paper electrophoresis that we have discussed since morning. Now comes the third part within the zone electrophoresis, your pulsed field gel electrophoresis. This is basically it's a technique separation of large DNA molecules, which is not possible with the help of your Agarose gel electrophoresis. Yeah, Agarose electrophoresis is not possible. Then we use pulse field gel electrophoresis by applying to gel matrix an electric field that periodically changes directions. So in case the periodic changes, what happen is we have a generally small fragments can find their way through the gel matrix in the normal case, but this is a large DNA fragments. For need in order to run them, like they could be sizes of 30 to 50 kilo base pairs, large fragments. So they should be run at the same rate and appear in gel as a single large diffuse band. So, but in order to find this, the difficulty, we have a periodical changes of field direction. So it's not going forward from top to bottom. It will be going zigzag way. So various we will provide the charge from the various ways. So the voltage is periodically switched among three directions. One that runs to the central axis also, and two that run at the 60-degree either side. Yeah, one on the center and two on the 60-degree size, and the times that are equal for each direction, resulting in the net forwarding migration of the DNA. So this procedure takes longer than the normal gel electrophoresis with the size of the fragments, but resolve the DNA that we are looking for very easily. So like this, as I discussed, you put 60 degrees Celsius watt on the central axis, and also from the side, from this way also and from this way also. Because of that, the resolution of your DNA might be possible because the sizes are quite big. If you run them like only straight way, so the resolution would be like some 2 or 3 DNA molecules will come together, and you can't able to find the good results. You might be having false readouts. So in that case, this pulse gel electrophoresis playing quite important role. I can show you one video about that, how it's done in the lab.
Pulse field gel electrophoresis, or PFGE, is a technique for the separation of very large DNA molecules. Under standard electrophoresis conditions, an electric field is applied in one direction on the agarose gel, and DNA molecules larger than 40 kilobases essentially move together through the gel, independent of their size. In contrast, PFGE applies an electric field that periodically changes direction. By introducing an alternating voltage gradient on a PFGE system, very large DNA fragments can be separated by their reorientation and movement at different speeds through the gel. Larger pieces of DNA are slower to align their charge to the change in direction, while smaller pieces are quicker to align, resulting in greater separation. Let's take a closer look at the equipment and conditions that will lead to successful PFGE. Here is the electrophoresis chamber, which contains the running buffer. The agarose gel should be well anchored in the middle of the chamber so that it doesn't float around.
There are multiple pairs of electrodes around the gel, set in a hexagonal grid. This hexagonal shape of electrodes allows for the directional switch of voltage. The voltage is periodically switched among 3 directions, one that runs through the central axis of the gel, and 2 that run at 60-degree angles on either side. The PFGE technique takes longer than standard gel electrophoresis due to the size of the DNA fragments and because the fragments don't move in a straight line. To prevent the agarose gel from overheating due to the extended period under the electrical field, which could result in a loss of resolution, a cooling module adjusts the temperature of the buffer. A typical PFGE program is run at 14 or 15 degrees Celsius. It is important that no air is left in the tubing to and from the cooling module. On the control module, the migration protocol can be entered and monitored. This includes the initial and final switch times, the duration of the migration, the angle of electrophoresis, and the voltage. The larger the DNA fragments, the longer the pulse times should be, and the longer the total migration time. The switch time is the length of time that the electrical field is pulsed in a single direction. For example, a 10-second switch time means that the electrical field will be pulsed in one direction for 10 seconds and then switch to another direction for 10 seconds. Samples with a wide range of DNA fragment sizes, such as NEB's PFG markers, can be resolved by ramping up a short switch time to a longer one over the course of the run. Most PFGE protocols are optimized for 6 volts per centimeter gel runs and optimized for a 120-degree angle. With any PFGE system, depending on the migration protocol, you can separate DNA fragments or molecules from 10 kilobases to 10 megabases. Here are separation examples of the lambda monocut mix from 1.5 to 48.5 kilobases, and yeast chromosomes from 225 to 1900 kilobases.
Okay, so I think we can make an end here today. We meet in 1 hour at 11. Just have your breakfast because you are awake since morning. I will also have my breakfast, have some rest, and then we see each other. We continue with the electrophoresis. So I will not close this one. So see you soon, students. Take care. We see us in 1 hour at it's 9. No, not in 1 hour. We see us at 11. 11, yeah. 11 in 2 hours. I have some work to do from my college, so in the meantime we'll do that, and then we will continue like the same. I will write in the Telegram group. Okay, see you soon.